Now we'll stick with the classic analysis resource, so the legacy site that's been around, because I'd like to invite Dr. Alessandro Sette back to present the analysis tools. So thank you very much, Alex.
Thank you, Nina. So what are the analysis tools? So the tools that Bjoern has showed you are mostly focused on making predictions for binding immunogenicity and so forth. There is another set of tools which are used to analyze epitope data in various different respects. And I'm going to show you around the part of the analysis resource that is devoted to the analysis tools. And you see here, you can reach them by the tab analysis tools. And you immediately see that there are two flavors. One are the tools that are listed just with their name. And then there is this other set of tools that have a flask. And those are, as the note underneath says, those are tools that are still a bit experimental and not quite ready for production. So they're still being used and still making changes here and there, but still they're totally usable. So, for example, the immunobrowser. The immunobrowser, you have heard about the immunobrowser capability earlier when we were showing you how the immunobrowser tools within a given query visualizes and essentially collates all the data related to a sequence mapping the immunogenicity density along with sequence. Now, there is also a standalone version of the immunobrowser. And so in this particular case, you can basically have your own data in the standalone, and you can have your own immunogenicity study that you want to combine, or you want to do a study with predicted epitopes or whatever. And this is visualized pretty much the same way the immunobrowser tool is operating from the main website if you run a query with the same type of outputs. Another tool, and throughout I'm just going to here briefly mention how the different tools work and what's the point, what's the goal. And for each of the tools, there is a lot of support in terms of referring to detailed examples or publication and so forth, but I'm not going to go into that level of detail here. Another tool, which is actually very often used, and it's one of the tools that is most frequently cited, is the population coverage tools. And so the population coverage tools answer a question that is often posed in the sense of saying, I have a bunch of epitopes that I know are restricted or bind to a set of different HLA molecules. What kind of coverage is provided by these peptides, knowing that they are restricted to these different HLAs, also considering that the different HLA alleles vary in frequencies in different ethnicities. And I've already showed some of this type of data, but this is the tool that is on the website, and it calculates the fraction of individuals that are projected to bind or respond to a given set of epitopes. And so here you need to enter your set of epitopes, what are they binding or restricting to, and the tool has the HLA frequencies and then calculates what is showing here on the right-hand side. The current tool has HLA from allele frequencies, not net. And the issue with this particular website is that is listing HLA frequencies based on the endogenous populations. So, for example, you might get in particular regions the frequencies of, so in Australia, of the HLAs in the Aboriginal populations, which are the ones originally populated Australia. But if you go into, doing a projected population coverage in Sydney, Australia, there's a lot more people there that have rather different ethnic heritage. So, and we'll get back to that issue in a second, because there is another set of HLA frequency that we can access that solves that problem. But regardless, what the tool does, for example, if you select a bunch of different population areas, we will calculate here what is the coverage defined by with those particular peptides, what fraction of the population binds at least one. And so in this particular example, you have that on average, this population are getting 75% population coverage. Or on here, you say these are covered by about, on average, 7 hits per donor and so forth. And then this also can be plotted, showing how many hits per fraction of individuals are covered and so forth. Now, as you're saying, this is an example of the kind of data that you get where you have different collections of peptides at different coverage around different general broad categories or worldwide. Another tool is the Epitope conservancy analysis. And also Alba alluded how we use conservation as an important parameter to, for example, select particular epitopes that might be of interest, or just calculate whether T-cell response directed against one particular region will be, for example, conserved in different isolates of a particular virus or bacteria and so forth. And so here the tool works by asking you to enter your sequence of interest in the sense of the epitopes, but you want to calculate degree of conservation. And this can be done with an adjustable sequence identity threshold. And this can be done when also you need, of course, to enter the set of protein sequences in which you want to calculate the conservation. And so here in this particular cases, we're looking at Lassa epitopes, and you have a number of different nucleoprotein epitopes, and then enter a number of different Lassa protein sequences. And in this particular case, we're entering also looking at the conservation of Lassa epitopes in this is GPC, but just to give you an idea of different arena viruses, different Lassa isolates, but also different old world arena viruses such as Mopeia or LCMV. And then, of course, another group could be other new world arena viruses. And here's a list of these different viruses. And the bottom line is that by doing that, you can calculate for different regions, with CEDAR's regions that Alba was showing you, how well conserved are these epitopes between Lassa lineage 4, which is one of the lineages that is associated with most human infections, but also there are other lineages. So you can ask, well, are these epitopes conserved across other Lassa lineages or other old world arenaviruses or other new world arenaviruses? And you see that the conservation drops as you move away from Lassa lineage 4, but there are some regions that are actually remarkably conserved across all different arenaviruses. Another tool is the epitope cluster analysis. So this answers a fundamental question that comes up often because you might have a set of peptides or epitopes that are largely overlapping with each other. And so you might say, well, okay, these are, 5 different sequences that are like 95% overlapping with each other, just that they start a little sooner or end a little later. And so how can I just cluster them to see how many groups of really epitopes are there? And there are many different ways to do that. And one approach is basically imposing that each epitope is found only in one cluster, so that you have non-redundant set of clusters, so to speak. Or you can say if this particular epitope is 70% homologous to one group of sequences, but is also 60% homologous to another set of sequences, then I can find the same epitope represented twice in 2 different clusters. And then there is one approach that is in between. And this is a tool that have very diverse applications. For example, you can use it to generate epitope pools, where you want to have only one representative for each cluster, or understanding cross-reactivity and so forth. And this is how the web page works, where you can put your sequences in, select the clustering method, and you will also hear more about this tomorrow. Another tool which is quite powerful is the rate tool. And essentially, the rate tool was developed taking the lead from classic genetics and odds ratio analysis. And what the tool does is guesses restriction, HLA restriction of epitopes based on known reactivity data. So you may know, for example, in the classic autoimmunity genetic analysis, that ankylosing spondylitis is very strongly associated with HLA-B27. Okay, so what does it mean? It means that people that have the disease have a much higher frequency of that HLA-B27 gene compared to people that do not have a disease. And in fact, the ratio of a frequency in these 2 different populations is what is commonly referred to as an odd ratio. So we reasoned that this could be used to actually infer restrictions. So in this particular example, these are a set of peptides. I don't even remember, probably mycobacterial tuberculosis peptides, but it doesn't matter. So this particular peptide and the DR beta 3 or 2 combination. If you look at this, there are these different outcomes. With A plus or R plus means that these are people that have that HLA allele and also responded, recognized this peptide. Then the A minus R plus are people that do not have this particular allele, and responded. These are people that have the allele, do not respond, and these are people that do not have the allele and do not respond. So there were 50 people that were tested for this peptide, and you can see that there are 6 people that responded to that peptide that had that allele, compared to 0 people that responded, did not have that allele. In other words, also you can look at the frequency, so that 6 people that have the allele responded and 18 people that had the allele did not respond. While conversely, of the 26 people that did not have the alleles, none responded. So based on this, you can calculate odds ratios and relative frequency, and then a p-value, which is a Fisher, exact Fisher p-value, which is not inferiorly correct at this point. And so this gives you some pretty good clues on some HLA molecules that may be restricted to different peptides, and then you can, for example, make those peptides and combine them with predictions. You can ask, does this peptide is predicted to bind to this allele? And you can make, for example, tetramers and so forth. And we found this very useful, as a matter of fact, to propose potential leads to actually produce tetramers for further testing. Deimmunization, this is a tool that is often used by people working with an interest of preventing immunogenicity. And this is done in the context of, for example, protein drugs, antibodies, or other drugs based on proteins that may have been engineered and so forth. They're injected into people, and sometimes what happens is that the patient treated with a drug develops a anti-drug neutralizing antibody response, or ADA. And that obviously is not desirable because it can lead to problems in terms of safety, or just basically the antibody can neutralize the activity of a drug. And so there is a lot of effort that is done to predict and potentially engineer proteins to remove sites that are potentially immunogenic. And this deimmunization tool does that. There's also a companion tool that predicts immunogenicity based on data, but we're not going to discuss that. And so here, for example, the tool predicted that this is a epitope that is likely to be a potential epitope recognized by humans based on its promiscuous binding activity. And then the tool generates all possible different single amino acid substitutions, and then calculates what is the predicted percentile binding of the single amino acid substitution peptide, looking for instances in which there is a significant reduction in binding capacity compared to the wild type. So, for example, here we'll see that we've done a significant loss of binding, where this was predicted to bind in the 10th percentile, but now this one is predicted to bind 3 times less, and so forth. Another tool is the PepSico tool, and basically this is a tool that predicts how easy or not is to synthesize particular peptides. And this is based on experience that has accumulated over the years on the ease to synthesize synthetic peptides. And so you can enter your sequences, and then you have a score that tells you how easy is this peptide predicted to be the ease at which this peptide can be synthesized. And this, for example, is a pretty bad score, which you can see this is a peptide that is made of many different hydrophobic residues repeat. And then this is historically it's hard to synthesize these peptides because the peptide sticks to the solid bead while it's being synthesized, and so it doesn't become accessible for the next amino acid to come in. Finally, another tool that has been generated is a tool that looks for data on whether that particular peptide is what is the estimate expression level of those peptides. And this is very important because apart from binding affinity, also the level of expression of a particular peptide is potentially play an important role in immunogenicity. Remember, we were discussing how binding is a necessary prerequisite, but not all things that bind are immunogenic. And expression level is one of the things that is in a dialogue with binding. So you can have something that is poorly expressed, but is very, very strong binder, and a similar level of immunogenicity may be achieved by something that is expressed at very, very high level. And this high level of expression is somehow compensating for a lower binding affinity. It's basically a simple thermodynamic equilibrium of the binding affinity. So this is to recap all these different tools that are available on the analysis tools section for immunobrowser, population coverage, the conservancy, cluster, rate, deimmunization, PepSISCO, and PepX. And with that, I will stop and go back to this.
Excellent. Thank you so much, Alex. We do have a few questions coming through, so if you would like to take a look at the Q&A, I'll give you some time, and then I'll also invite Bjoern back to the virtual stage to kick us off with our first question. We are running a few minutes ahead of schedule as well, so there is some time, but Bjoern, take it away.
Okay, awesome. So starting with the very first one from an anonymous attendee. So when is NetMHC Pan version 4.2 becoming available in IEDB tools? The quick answer is the next scheduled release is for the version 4 release, which is going to be in the beginning of 2026, and then right after the 4.2 is coming out. So this would be probably mid-year 2026. But I think more generally, I should be pointing out that this is a general thing, right? As tools become available, typically after they're being published, we are getting them implemented on the IEDB site and then making them available for users. And so there is typically a year time lag, and that is something that I'm not particularly proud of. I would love to make this faster, but this is where we are right now. Alex, you want to go next?
Yes, is a question, is a good binder always a good immunogenic peptide? And the answer is no. So there are many reasons for that. And as I was saying, one is that T cells recognize a complex between a peptide and MHC. But if there is no T cell that is willing to recognize that complex, then the peptide is not immunogenic. And as we were mentioning earlier, there is a process by which the body eliminates or keeps at bay T cells that react against the cells. And that is one process is central tolerance in the thymus, and the other is peripheral tolerance. So that process may wipe out or greatly reduce the immunogenicity of peptides that are perfectly good binders. And then another factor that we understand is playing a role in influencing immunogenicity is the level of expression of that sequence, that peptide, in the particular virus or bacteria or the body. And obviously, the higher the expression in a virus or a foreign entity, the higher the likelihood that this will be recognized because more complexes are formed. Another element that influences immunogenicity is the degree of conservation of that sequence across different species. And Bjoern and I did some studies a long time ago where we were showing that, for example, this matters in the case of allergies. And why does it matter? Because you may very well be allergic to Timothy grass. And so when you breathe Timothy grass, you sneeze. But then the next day you get in your nose ryegrass pollen, and then next cedar pollen, and then next. So there's lots of grasses of trees that have pollen. And so if there is a sequence that is conserved across different pollens, you get more stimulation. And so to understand that, there's, for example, the conservation tool does answer a little bit of that, but this is also important in determining the immunogenicity beyond the binding. But binding is certainly a fundamental variable in determining immunogenicity. And if a peptide doesn't bind, nothing will happen.
Thanks, Alex. I'll take the next one from another anonymous attendee. Is the trained dataset valid for all epitope types, like infectious versus allergy versus autoimmune, or should we change some parameters to adapt the prediction accordingly? So I would say for MHC binding and related factors that are involved about like, does the peptide actually get presented by an MHC molecule and the affinities of that, there is no knowledge at that point of the disease that is associated with. We do not think that there's a reason, and we have not seen any evidence that there's any reason to adjust that. Now, if you have a more complex pipeline that does things like eliminate things based on self-similarity or so, that will definitely be extremely dependent on what are you doing. Autoimmunity is defined by that the things that's being recognized are self-similar in infectious agents. We and others have shown and tried with that there is a evidence for negative selection, so that you actually have less recognition of peptides that are similar to self. So I guess it depends on the question and depends on the tool. Sorry to not be more helpful. Alex, you have another one?
Yes, there is. A Harley says, where did he go? Okay, yeah. For each tool, it's possible to glean the data it was trained on. Is it public? Accuracy of the model, including immunogenicity and others. So in general, we try to make that information available and disclosed. And in general, most of the tools, there is a paper that describes the tool and published in peer review. And when that is available, you will find it in the documentation of the tools, and then you can drill down, and that will say, oh, we developed this tool, we trained this on this data set, and then we validated with this other data set. So in general, if that information is available, is provided in the documentation accompanying each different tool, which obviously each tool is developed and trained on different data sets by its own nature. Bjoern.
Thank you, Alex. Next one I have is Björn. Thank you very much for your talk. For another anonymous attendee. Um, I'm going to rephrase this a bit. Um, So at this point, it feels like where the egg comes from scenario, and I think it's like a chicken and egg thing about there's the standard MS assays use MH and that MHC pan to affirm binding between polled peptides and the HLA allele versus then the same net MHC pan is trained on that data. So how does the timeline of this work? Like who's the chicken and egg thing? Where do these things come from? So that is a real issue. I mean, to be clear here, like the issue is you have data that is being uh, published where tools that are being used in the that have been developed using IDB data are used to assign like restriction or filter out peptides and saying, well, we don't know what this one is about, and we leave it out, and that is becomes then a kind of a loop that can be very negative. So we very much encourage people to always publish the entirety of the data, like say in MHC ligand dilution, including the ones where they say they can't assign restriction for, because that might be exactly that you might have a peptide that right now can't be assigned because there's a missing information essentially about a specific allele or so. So overall, the versions are time-stamped of the tools that we have. And then you should look at the data sets that get published afterwards. We have been trying as much as possible to look at the raw data, specifically MHC ligand dilution, and then reassign restrictions in data sets where you have well assigned alleles present in the donor. So these things work, but there is an issue of like confirmation bias, if you want. At the same time, actually, there's plenty of papers that are identifying interesting peptides that do not fit the standard predictions, and following up on them is interesting as well. So that's not a very satisfying answer, but I hope it's somewhat useful. Alex, please take the next one and be better than me.
All right. So the question that I'm going to answer is whether does PepsiCo, I mean, whoever came up with his name should be hanged. But anyway, does that tool was it that trained on real data, or is it purely theoretical? And this is actually a good example to go back to what I was saying, is the data on which the algorithms and tools been trained on available, all disclosed. And if you go to the PepsiCo, there is a and you look on the thing, there is a tab that says reference, and you click on the tab and you will get the paper that we published describing that tool. But since obviously you don't need to go and read it right now, but basically what happened is that we synthesize thousands and thousands of peptides. And also, as a quality control, we run mass spec. And so we see that sometimes the peptide is bad. And that created a very large database of good synthesis and bad synthesis. And that was what was the tool based on. And there was another related thing. Does the paper list synthesis condition? This is all FMOC chemistry, and certainly the paper doesn't say, oh, if this is a sequence, you should use a different chemistry or different strategy for synthesis. This is it's not meant to be a replacement for a good peptide chemist to solve problems. Yeah, Bjorn.
Okay, so I'll answer Another anonymous attendee, do you plan to revise the reference set for percentile rank calibration for MHC predictions? The answer is yes. Every time we're doing a new release with new tools, we are recalculating. If there's specific issues you run into, please let us know through the help desk. The next, right after, is the next gen tool resource mainly a clearinghouse for tools developed by others that have names, like NetMHC Pan, or are some developed by internal IEDB groups? If so, how can we tell the difference? Actually, NetMHC Pan is developed by an IDB group, and that is Morten Nielsen, who is a subcontract on the IDB, and all the development that has been going on with that has been co-funded by IDB tools. So that is, but the names, when you say have names, we kind of give names like PepSisco, not PepSico. In an attempt to make them more sellable. So many of the tools that we have right now are maintained by people involved in the IDB. But again, MHC Flurry, for example, is a good example of something that comes from completely outside, is generated and maintained by other people, but we are providing a stable interface to it. Alex.
There was one question: where does the PEP-X expression prediction based on? This is not prediction; is extracting data from different publicly available sources. Correct, Bjorn?
Yeah. So then I think I have maybe the last one. I don't know, Nina, where we're running, but there's like two related questions. Anonymous asks, what is the main reason in your view that people do not submit their tools to IDB so far? And Mauricio asks, Bjorn, could you please provide your perspective on the appropriate stage in the process at which user-provided tools should be made available for the IDB? So, first of all, I mean, we've been trying to reach out and get people to submit. So any recommendations on how to make it better would be very welcome, and you can email us or so. My personal gut feeling is, I mean, often this is like an afterthought. Often, like you want to submit your paper with your tool and get it out there, and then the level of effort you have available drops. And providing us a tool for implementation will require still some effort, right? That we have to work with you on getting the tools running or so. And very often, then the level of effort available is not there. And then I think people are dropping off and they don't necessarily see the benefit. There's also often a mismatch between the PI and the person developing the tool of what the interests are. But again, like we would love to do more of this. I mean, I remember being a PhD student and thinking about like, okay, what would it take to get a tool on the PDB side? And I thought this is impossible. And then when I met the PDB folks and they were so excited about it, it was it was a surprise. I think one of the other things is people don't think it's possible, but it's very possible, and we would like that. And then for Mauricio's question of when is the appropriate stage. Again, like we do not want to take credit for stuff, so I think it should always be after a tool has been published and is peer reviewed and is yours and everything, and then we would like to make it available when it's like in a stable form where we can integrate it on the website. That would be the best thing. And actually, also pointing out one other reason I forgot about that for not implementing. Some tools require massive computational effort, and there we simply can't commit to it. It must be we must be able to run it. So that's one of the exclusion criteria. We can't host tools that are going to lock down our server for days to run one prediction. I hope that's obviously understandable. That's it for me.
Excellent, Alex. Any other questions for you?
No, I dismissed a number of questions that are all related to where to get the expression levels come from as a public database. It's not that I didn't like the answer, but I think we answered it.
Absolutely.
Yeah,
There's
I
one.
think that's about it.
Perfect. And the question from Clarissa, I am getting some updates, but we will confirm tomorrow on day 2. But Clarissa said, awesome presentations. I want to know what is the maximum number of protein faster sequences that I can use at the same time for prediction of class 1 epitopes in the IEDB site, as she's had some challenges with that. And we believe it's actually a character limit of 10,000 rather than a sequence limit. So that's the current information I'm getting, but we will double confirm with you guys tomorrow when we go through the T-cell class 1 tool suite in a lot of detail. So thanks for that question too. And then with that, we're doing pretty well for timing. So thank you so much, Bjorn and Alex, for tackling our last Q&A of the day. Thank you both.